Neurons are excitable cells because their membranes are in a polarized state. Different types of ion channels are present on the neural membrane. These ion channels are selectively permeable to different ions. When a neuron is not conducting any impulse, that is resting state, the axonal membrane is comparatively more permeable to potassium ions and nearly impermeable to sodium ions. Similarly, the membrane is impermeable to negatively charged proteins present in the exoplasm. Consequently, the exoplasm inside the axons contains high concentration of potassium and negatively charged proteins and low concentration of sodium. In contrast, the fluid inside the axon contains a low concentration of potassium and a high concentration of sodium and thus form a concentration gradient. These ionic gradients across the resting membrane are maintained by the active ionic gradients across the resting membrane are maintained by the active transport of ions by the sodium potassium pump, which transports 3 sodium outward for 2 potassium into the cell. As a result, the outer surface of the axonal membrane possesses a positively charged while its inner surface becomes negatively charged and therefore is polarized. The electrical potential differences across the resting plasma membrane is called as the resting potential. When a stimulus is applied at a site, for example, as you can see on a point A on the polarized membrane, the membrane at the site A becomes freely permeable to sodium ions. This led to a rapid influx of sodium ions followed by the reversal of the polarity at that site. That is, the outer surface of the membrane becomes negatively charged and the inner side becomes positively charged. The polarity of the membranes at the site A is thus reversed and hence depolarized. The electrical potential differences across the plasma membrane at the site A is called the action potential, which is in fact termed as a nerve impulse. At sites immediately ahead the axon, that is the site B, membrane has a positively charged on the outer surface and a negatively charged on its inner surface. As a result, a current flows on the inner surface from site A to site B. On the outer surface, current flows from site B to site A to complete the circuit of the current flow. Hence, the polarity at the site is reversed and an action potential is generated at site B. Thus, the impulse, that is, the action potential generated at site A arrives at site B. The sequence is repeated along the length of the axon and consequently the impulse is conducted. The rise in the stimulus induced permeability to sodium ions is extremely short lived. It is quickly followed by a rise in permeability of potassium ion. Within a fraction of a second, potassium diffuses outside the membrane and restores the resting potential of the membrane at the site of excitation, and the fiber becomes once more responsible to further stimulation.